Hello, so my name is Giovanna Onofrietti. My science research was on the inhibitory effects of cannabidiol and xylitol on pathogenic oral bacteria. So in my introduction, first off, we have periodontal disease. Now, periodontal disease is the destruction of connective tissue and dental bone support, and it has many inflammatory host response secondary to bacterial infection. Periodontal disease is actually the major cause of tooth loss and most common oral diseases in the mouth. Its main biological factors, which means things that can lead to periodontal disease, is bacterial plaque, microbial byproducts such as different brands of toothpaste that you use in different things, and of course the host immune response. I just gave a diagram of the periodontitis and what it would look in healthy gums compared to periodontitis. You can see how much damage builds up when you have periodontitis and how these things affect it, and the bacteria affects it in the mouth. And you can see here the healthy gums and the enamel and what it would look like healthy compared to periodontitis and how much it ruins the tooth and the enamel and the bone structure. Moving on is Fusobacterium nucleatum. This is one of the bacteria I used to test in the project where I move on early on, but it's actually one of the most abundant bacteria in oral cavity, and it has a big influence on producing these dental caries. And of course, it is a gram-negative, non-spore forming anaerobic species, and it's known for its prevalence in oral biofilm, and that means just oral plaque and how much it builds up over it and how much that makes an impact on the tooth and how much that bacteria actually builds up on the tooth. Moving on to dental caries. Now, dental caries is actually one of the most common infectious diseases in humans, and things that cause this dental caries, of course, is poor oral hygiene, carcinogenic microorganisms and different things that you eat, and different consumptions of refined carbohydrates can really affect these dental caries. And of course, poor oral hygiene is definitely something that causes it. And it's actually also, dental caries are one of the most common diseases in the mouth between teenagers and adults. And I just gave an example and a diagram of dental caries and the enamel and the different surfaces and what actually affects, and how you can see in the middle it affects the actual bone and the pulp chamber and the blood vessels and how deep it goes into the enamel. Next is Streptococcus mutans. And actually, Streptococcus mutans are actually the bacteria that cause dental caries the most, and they're associated with this tooth destruction, and it causes impaired speech and multiple infections. Streptococcus mutans are the bacteria in the mouth that mostly cause dental caries. Next is cannabidiol. This is one of the two natural things that I chose to work with my project, as I will get into that later when I discuss, exactly what I did. But cannabidiol is actually one of the most numerous phytocannabinoids or active compounds found in the Cannabis sativa plant. There's actually a lot of research going on with CBD oil and different things, and how it is the most potent and purest form of the hemp plant, containing upwards of 90% cannabidiol with no psychoactive effects, and is both tasteless and odorless. Now, I just want to do another continuing with cannabidiol. Its demand is rapidly rising as scientists discover potential reduces to seizures, pain, anxiety, and other things. Its effects could be potentially beneficial to different diseases like Alzheimer's diseases or some cancers, so we're definitely looking into that, and many scientists are too. And this form is actually the CBD hemp product that is 100% free of THC, meaning that cannabidiol is free, leaving pure CBD, and there is no THC in it, which many people got confused. And the other thing I worked with besides cannabidiol was xylitol. I actually worked with xylitol in the past in the mouth, and that's why I consider it, because I know that it works, and cannabidiol does lower the pH it does not lower the pH of the oral cavity, and it has the ability to reduce plaque on the tooth, which means it has the ability to reduce oral cavities, as I worked with previously, but I want to work with it again because I want to see the effects of it, combined with something else, of course. So my hypothesis states, if Streptococcus mutans and Fusobacterium nucleatum are exposed to plant derivatives, cannabidiol and xylitol at various concentrations based off the minimum inhibitory concentration, then the plant derivative mixture with sub-minimum inhibitory concentration would create the largest zones of inhibition for both pathogens than any of the plant derivatives individually, because cannabidiol and xylitol combined, even at lowest concentrations, will produce synergistic effects greater than the sum of individual effects. So basically what I'm trying to find is by both them together will create a bigger effect to get rid of these dental caries than combined individually. or them just individually. So my methods, I kind of described what I use in the methods too, like my materials. So I use petri dishes with 5% sheep blood for the Streptococcus mutans, and I use this because 5% sheep blood agar is actually better for bacteria to grow on. And I had a total of 132 trials. To do this, I used the streak method to cover the blood agar plates with the S mutans and then the F nucleatum separately, and then they were incubated at 24 hours at 37 degrees Celsius. Then, after I did them individually, I did the mixtures, which they were 50-50 concentration of both the CBD and xylitol to see their synergistic effects together. And before extract were applied, sterile discs were applied to the petri dish for the Kirby bucket method. 6 trials were performed for each concentration of the CBD and xylitol, and they were performed together. Plates were examined after 24 hours of incubation, and the diameter of zones of inhibition were measured in millimeters to see which one had the bigger zone of inhibition. And as I said before, tests were done 6 times to minimize test error to make sure that they were correct for a total of 132 trials. So here in my data chart, you can see, not very clearly, but you can see the cannabidiol, xylitol, and the cannabidiol and xylitol mixtures, and all their data, and how many in the micropipette, how many concentrations were used in the different amounts, and how much they grew in the zone of inhibitions were measured. And I did a positive and negative control, and the positive control was 1% sodium fluoride, and the other one was chlorhexidine glucose, and the negative control was just distilled water, as they did not grow. So for my bar graphs, you can see the average zone of inhibition of Streptococcus mutans, and then the average zone of inhibition of the Fusobacterium nucleatum. You can see how in both graphs, cannabidiol was significantly larger than xylitol, and how it had more of an impact just alone on both bacteria than the xylitol did. But, as you can see better in my bar graphs of the average of zone of inhibition of Streptococcus mutans and Fusobacterium nucleatum, that the synergistic effects of both cannabidiol and xylitol mixtures had a great impact just together than individually, proving my hypothesis correct that the synergistic effects of cannabidiol and xylitol mixtures worked better and will prevent dental caries rather than just individually. For the ANOVA, I decided to do an ANOVA statistical analysis to prove my data was statistically significant, and you can see that in my ANOVA chart that my data was very statistically significant, and it showed that, ANOVA, your data has to be significant below 0.01, and my data was very significant, proving it was correct. Now, in my discussion, I just gave some of the highest concentrations, and you can see that my highest concentration between the cannabidiol and xylitol concentrations, where P is less than 0.0042 and P is less than 0.0059, which means it is very significant and was correct. And the discussion continued. I did the individual verse mixtures of cannabidiol and xylitol and the mixtures of both of them, and they were very significant with a p-value of less than 0.001 and a p-value of less than 0.0144. So in conclusion, the synergistic effects of the CBD and xylitol mixtures from both bacteria in all concentrations had a larger zones of inhibitions in comparison to each concentration individually. So this proved my hypothesis correct. The synergistic effects of both of them did have a bigger impact on the dental caries and will prevent them rather than just separately. And that's what I was trying to figure out. So the S mutants and F nucleatum are susceptible to CBD and xylitol mixtures, even at a sub-MIC. Now, sub-MIC is a microorganism inhibitory concentration, that is below it. So I used a lesser amount and even showed how big of an effect that was. And the inhibitory effects of the plant derivatives could enhance the antimicrobial efficiency against both these common oral disease pathogens like dental caries and periodontist disease, and it can have a really big impact and can really improve different dental cares and different way we treat them, and can really have a big impact. And of course, for future research, I really want to do more of this in mouth, maybe by using more natural substances or different things, or looking into more deeper dental problems and different dental diseases that have a bigger impact than dental caries and periodontal disease, that are more serious and more severe to the mouth. And that is really what I want to work with, because I'm so interested in the mouth and how to get rid of these dental caries that can really hurt people and really harm people in their mouth. And that's really what I'm striving for in this, and to use different natural substances, because some of our toothpaste have cancer-causing agents or different things that are not so healthy in your mouth. So by using these natural agents, we can be able to put stuff in our mouth that will not harm us later in life. Thank you.